Yeah, hi everyone. I'm Laura Luebbert. I'm a Eric and Wendy Schmidt Fellow at the Broad Institute and also a Future House AI for Science Fellow. I'm mostly a postdoc in the Sabeti Lab, though I'll be talking today about work from my PhD in the Pachter Lab. In the Sabeti Lab, I'm very involved in the Sentinel project for pandemic prevention. I won't be talking about that at all today, but if you're interested, feel free to reach out. And as Hannes mentioned, this might be a bit more biology heavy than some of the other lectures, but I try to make it accessible and exciting, hopefully, for both the more biology-oriented people and the more male-oriented people. And if anything's unclear, feel free to interrupt me at any time. All right. So the paper I'll be talking about was on the cover this January in Nature Biotechnology. So I just wanted to give a shout out to Christine, who's a student in the Sabeti Lab who designed the cover. I did also want to give you a bit of a timeline behind this paper. So the first version of this paper came out in 2023. And honestly, not that much happened since then. The reviews were really smooth. There haven't been major changes to the technology. But yeah, it only was printed in Nature Biotechnology this January. So I won't go into a rant about scientific publishing, but just wanted to give the timeline. I'll also give you a bit of an overview of what happened in the field since this came out in 2023. All right, so when I first made the first version of these slides in 2022, there were two things I was really excited about. One is this image came out, which was the deepest infrared image at the time of our universe, taken by the James Webb Telescope. And I'm going to use that image to put the second thing I was excited about into perspective. So this image, according to NASA, is like holding a sandcorn at arm's length. And so this image and all of the stars within it are just the sandcorn in our night sky. If we were able to see our entire night sky, we would see between 10 to the 22 to 10 to the 24 stars. I'm going to use that number to put another number into perspective, which is the total number of individual viruses on Earth, which is 10 to the 31. So about 10 million viruses on Earth for every star in the universe, which is insane, which also makes them the biologically dominant entity on Earth by numbers, not by weight. I tried to make these circles to scale, but it was impossible. I couldn't make it small enough. Okay. All right, so today I'm going to be talking about one specific realm of viruses, namely riboviria or RNA viruses. At the time, we knew about 200,000 taxonomies of RNA viruses, and RNA viruses are very important to humans because they cause the majority of common human viral diseases. We're all really familiar with coronaviruses after SARS-CoV-2, but also dengue, Ebola, Hep B, influenza. These are all RNA viruses. So it makes sense that we would want to study them. And the way that I was studying them mostly was through a technology called single-cell RNA sequencing. So I just wanted to give a quick overview of single-cell RNA sequencing, and then I'll go back to viruses in case people here are less familiar with the technology. So in single-cell RNA sequencing, you take a tissue of interest, you take a piece of it, you dissociate it into individual cells, and then you measure the RNA in each individual cell. And what you get back is literally a text file with a bunch of A, T, Cs, and Gs. And from that, you try to make sense of that in the way of creating a matrix that is a cell by gene matrix. So for every cell, for every gene, we knew whether the gene was expressed or not and how highly it was expressed. And the way we go from text files to the cell by gene matrix is by aligning our ATCs and Gs to a reference to try to figure out which gene they corresponded to. And we could use this technology to similarly look for viral genes, for example, that are also picked up in the RNA, even of hosts of humans. The problem with that is that, so looking at this step where we have to align to references, going back to our RNA viruses, this is the number of curated NCBI reference genomes that we had for RNA viruses. And these slides are definitely a little outdated. This number has significantly increased over the last 2 years, but overall, this is still definitely a problem. We don't have high quality reference genomes for all the viruses we'd want to be looking for. And in addition to that, if there's a new virus, for example, we wouldn't be able to look for it even if we're picking up the signal.
Can
Hannes,
we
did you
go a
have a
slide back?
question? Oh, yeah.
Yeah. So here on the previous slide, we now have this tissue dissection. And is this then what we would want to also do to a human and then see if the tissue or the RNA-seq of that human matches up with the RNA-seq or something in that RNA-seq matches up with another virus and then see, aha, this patient or we diagnosed him with having this virus?
Yeah, so you could look at the human gene signal. I think that's what you were asked. Like you could look at the actual human genes or human RNA that you can find in the sample and see if there's some indication of a viral infection. You can also actually, I mean, the viruses are inside these cells and they're replicating and they're making viral RNA. So if there is
Yeah.
an active infection at the time of sequencing, you actually measure the viral RNA as part of your RNA signal. And so the challenge is to correctly identify that in your signal and identify it as viral.
But what we want to do in the end is to detect whether in some cells there is a virus or not.
Yeah,
Yeah. Okay.
yeah, exactly. Yeah. Yeah. And then in addition to that, we also have what the human genes did in that same cell.
So what is the single cell cDNA library preparation?
Oh, so basically the schematic is of a microfluidic device, and what happens is that every cell gets encapsulated into a droplet, and then in that droplet, we dissolve the cell wall to get to the RNA, and then you can't just measure it right away. That signal is too weak. You first have to replicate it,
Ah,
and that's what makes the cDNA library or copy DNA library. It's like copies of the RNA.
okay. And like so we we somehow because the hole that we press the cells through is of a certain size and only one cell fits through it at a time. It goes through
It's
there and then
It's actually, yeah, it's the size of the tube. It's actually also, it has a lot to do with the flow rate as well. So this is like a whole other field of like the biophysics of these microfluidic devices. But that's just
okay. And
aside. Yeah.
then we have a droplet and inside of the droplet we lyse it and do the replication.
Yeah. Yeah. That's how you
Okay.
get the single cell resolution. Yeah. It's quite crazy that it works. Yeah.
And how do you, and doing the replication means you put in some additional proteins that do the, what's it called? Something like PCR?
Basically, yeah. Yeah, you basically do PCR with a universal primer. So there's different technologies, but the simplest way is to do just a primer that should, in theory, work for every RNA molecule. And then you also barcode the cells so that then when you actually do the sequencing, you know which cell each RNA molecule came from based on that barcode that the cells were
Okay,
given in
because
this
you
lab vector.
then throw it like everything together in one soup when you do the sequencing instead of doing it person. Yeah, okay, okay.
Yes. Yeah, exactly. Yeah,
Makes sense.
great. All right.
But now we only have a few, and now we have our sequences, and then we want to align them to reference genomes, and then we see, aha, it finds no alignment, therefore it's not a virus, or we find an alignment, therefore it is this virus. But we might be missing many viruses because we only have 5,000 instead of the 186,000
Exactly.
reference
Yeah,
genomes.
exactly. And if you
Okay.
don't know what virus you're looking for, it might not even occur to you to align to that reference genome, right? And I'll talk about aligning if you just wanted to align to all of them, just to see. I'll talk
Uh,
about why that
yeah,
is computationally a challenge. But yeah.
nice.
All right. Okay. Thank you for the questions. This is great. Yeah, so overall the challenge is we just don't have enough reference genomes for viruses. Um, so what we do have in 2022, a new database came out called the PalmDB, and the PalmDB has 300,000 sequences for RNA viruses. So I'm going to tell you real quick what the PalmDB is, but the whole premise of this paper was, can we use the PalmDB as a reference when we're looking for viruses instead of relying on individual viral reference genomes? So what is the PalmDB? Basically, it is the result of a huge effort by Robert Edgar and Arden Babayan, who's at U Toronto. And they searched the entire sequence read archive, which is this massive database of sequencing data, where basically all scientists dump their sequencing data there when they publish. And they searched 5.7 million samples. So these are globally and ecologically diverse samples for proteins that look like a very typically viral protein. So that protein is called the RNA-dependent RNA polymerase, or RdRP. And this is a protein that basically only RNA viruses have. So we know if a protein looks like an RNA to RNA polymerase, it's probably an RNA virus. You can think, so this is kind of similar, like we humans, we have our DNA to RNA polymerase, but we don't have any RNA to RNA polymerase. We never make more RNA from existing RNA, but RNA viruses do. So this is a great protein because we can look for it and we'll know, okay, that looks viral. And they searched this entire huge database of all the sequencing that was done in agriculture, in humans, in pharma, in research, in dev bio, in infectious disease research for anything that looks like an RdRP. And then they made this consensus database of almost 300,000 RdRP sequences that represent all, or are supposed to represent all RNA viruses that were ever sequenced on purpose or by accident, and then deposited in this database. And so the cool thing is that we can now create kind of a reference genome for all of these RNA viruses, covering over 100,000 RNA virus species, but it's a relatively small index, so it's less than 600 megabytes. Versus if you wanted to build an index of just our 6,000 reference genomes that I talked about before, that would already be over 100 gigabytes. So very computationally expensive to do. All right.
Okay.
Yeah.
I don't quite understand what's going on. So we know that, or we have this database where we somehow leverage the knowledge that only RNA viruses replicate RNA itself rather than replicating and having mutations in DNA and translating those to RNA. And this PalmDB now, what does it allow, or what does it collect?
So it basically, it looks at all of these sequencing samples and it checks, did we ever on purpose or by accident sequence an RNA virus? That's the main goal. And the way that they did that is by looking for any sequences that look like they encode an RNA to RNA polymerase. So this protein has like very highly conserved signatures called the PALM motifs, hence PalmDB. And you can actually,
So
mhm.
we do whole genome sequencing, and then
Um, this is RNA sequencing, important for so it's mostly focused, it's mostly RNA sequencing. Yeah. And
um,
it's similar to technology to what I just showed. These are mostly bulk RNA sequencing samples. But you can just think of it as any lab anywhere in the world did some RNA sequencing for whatever reason, and then they made their data public. And maybe they were studying viruses, maybe they were studying, I don't know, like agriculture, something totally unrelated to viruses, but they happened to have an RNA virus in their sample, and they found it by looking for this RNA to RNA.
yeah, but
Right.
I just thought the way it works, we take like a piece of, like we always know that the thing where we find the RdRP in, it is also all the other sequences that we have in the same sample are from the same cell or from the same species at least. Or
Um,
not even cell, because sometimes it's from a virus.
yeah, so you would have a sample like, human sample. It's going to be mostly human RNA, but there might be viral RNA in there. And if there is, you could try to look for, if you have a sequence in there in a human sample or in a mice sample, a corn sample, whatever, um, if that sequence looks like an RNA to RNA polymerase, it probably came from a virus that was infecting or it was present for whatever reason in that sample at the time of
Yeah,
sequencing.
I understand that, but my confusion is now just we find that there, okay, like this is the sequence from start to end, and now it's highly conserved, so it's the same across all the different viruses. And
Uh,
now we find that there's the RdRP in my soup of human RNA sequences, right? And
yeah.
now like I can't just go ahead and now pretend that all the other RNA sequences in there are viral. They're human, most of them.
Yes. No, that is absolutely correct. Yeah. Most of them are going to be human.
So why
But
is it useful to detect this RdRP?
um, because so the RDAP, the palm domains are conserved enough that we can recognize the RDAP through the domains, but it's still different enough that we basically, if you take the whole RDAP sequence, you can distinguish between viruses.
Ah,
So
okay.
now that we have this PalmDB reference, it kind of gives us like a global reference for all RNA viruses, if that makes sense. So
But
even though you
only ever of the RdRP.
Yes, only based on the RDIP. And there are methods, you can try to do assemblies and try to get, longer sequences. So you can try to figure out actually like try to get the whole genome of the virus. Depending on the sequencing technology, that is easier or not so easy to do. For single cell, you basically cannot do that. For most bulk RNA sequencing, you can. But in this case, we're really going just based on the RDIP. Yeah. Does that make sense?
Yep, yep.
Okay, great. Awesome. Okay, cool. Yeah, definitely like keep asking questions. This is great.
I have a
Um,
question.
oh yeah.
So, what is a genome? Because the NCBI is much bigger than the RDRP, and I kind of don't understand why. Like, what is genome?
Why we would
No,
compare? Oh.
like, what's the two data set different in what way?
I
Um
don't. Go ahead.
The
yeah,
like we have two data set here, the NCBI and the RDRP, and what's the difference between the two?
oh,
Why? Yeah.
why yeah. So why is this so much smaller than
Yes.
the NCBI? It's mostly because so the NCBI is the whole genome versus this is just the RDIP. That's why it's smaller. Um, you could take just the RDIP sequences from these viral sequences and end up with a smaller database, but you would still only cover these like 6,000 or less viruses versus the over 100,000 that we have here.
And what is genome? Sorry.
Oh, okay, wow. I don't know where to start.
What is what? What's the question?
What is
Genome.
what is the genome
Okay,
of the
like
virus?
all the DNA in a species. Is that a good definition?
Yeah, I think that's
Oh,
that's a fine definition. Yeah, that's like it makes up the core of the virus, and it can have different architectures depending on the virus. But this right here, that is the genome of SARS-CoV-2, and the RDIP is one part of it.
oh, I
But
see.
it's the piece of DNA that encodes the virus, basically.
Yeah, thank you.
Of course. All right. Yeah, sorry. I now I wonder if I should have given more background.
No,
So yeah, please
it's
keep
good. It's
keep
good.
asking questions. All right. Um, well, I
Laura,
just wanted, oh yeah,
oh yeah. I also had a question.
yeah.
So I was wondering if people do pangenome graphs for these sequences because they seem to be at feasible lengths as well. And then you could maybe do sequence to graph mapping, etc. So yeah.
Laura, when you answer the question here, can you maybe also explain the meaning behind the question?
Yeah. So if I understand the question correctly, so you want to build a graph from the RdRP sequences,
Yeah.
is that right? And then sequence to graph mapping. Yes, so that's mostly done in terms of clustering. I mean, if we have like most of these RdRP sequences, we think they're RdRPs based on what they look like, but we don't know which virus they belong to. And so we estimate taxonomies by doing basically that, by trying to cluster them based on similarity, building trees from that. And then we just kind of draw circles and we say, okay, all of these, we're calling them, they're kind of similar to coronaviruses. So we're gonna do estimated taxonomy as these are the coronavirus RdRPs. I'm not aware of any sequence to graph mapping in terms of actually finding the sequences. I don't know how it would compare to, so the technology that I'm talking about today is a pseudo-alignment technology. The tricky thing with sequencing data is that you always need to do something that is very computationally efficient, because we're talking about mapping millions of sequences very quickly, or even billions of sequences with modern sequencing technologies and like current sample loads. But it's totally something you could do. Yeah. Cool. Awesome. All right. I mean, why do we even want to look for viruses globally in sequencing data? I think after SARS-CoV-2, I don't need to explain it too much, but generally, just by their sheer number, you can imagine viruses are floating around everywhere. Sometimes they contaminate laboratory reagents, which can cause problems in research, but it can also cause problems in healthcare. Like in the 1950s, a vaccine was contaminated with the carcinogenic virus, which obviously was really bad. And we're learning more and more that viruses are involved in diseases where we previously didn't expect them, with Epstein-Barr virus and multiple sclerosis, which recently came out. And more, it looks like there are viral roles in neurological diseases like Alzheimer's and Parkinson's as well. And also, I mean, there was the huge discussion around where SARS-CoV-2 came from. So overall, we're just really bad at keeping track of viruses, even those that can cause severe disease in humans. All right. NASA has a mission called NEOWISE, or they had a mission. They actually recently canceled it, unfortunately, but the analogy still works. So the NEOWISE mission was tracking asteroids around Earth and just automatically flagging any asteroids that could potentially become dangerous to Earth. And so I think we need a NEOWISE, but for viruses, for tracking viruses globally. And this is also very much something that the Sentinel mission in the Sabet Lab is working towards. So this is just a very, very small piece of that. But what tools do we have for viral NEOWISE? So there's a ton of tools that you can use to look for viral sequences in sequencing data. And again, this list is outdated. There are more tools that came out since I made these slides. One of them is Luebbert that you might be aware of, because it's more of a machine learning method that should absolutely be in this list. But all of them can use NCBI reference genomes to look for viruses in sequencing data. Very few of them can use the PalmDB, mostly because the PalmDB is an amino acid reference. So if we want to use the PalmDB as a reference, we need to do something called translated search or translated alignment, where we align nucleotide sequences like RNA to protein sequences, amino acids. The other thing is I wanted a tool that is able to retain the single cell resolution. So you have your text files with your ATCs and Gs. You don't just want to know which gene is this. You also want to know, you also want to keep track of which cell did it belong to. And that actually gets quite complicated with single cell sequencing technologies. And you do very much need specific algorithms for that. And most of these tools cannot or don't do that. They were not built for that. And lastly, I'm very passionate about software accessibility that can come in like many forms, but here I was mostly looking at, is there documentation for the tool? And does the tool actually install if you try to install it without spending hours trying to debug it? So we want a tool that is actually usable. Otherwise, it kind of defeats the point. All right, so when PalmDB came out, and I wanted to use it as a reference for my single cell data, we realized, oh, there isn't a tool that checks all of these boxes. So I decided to build it. Did you have a question, Hannes?
Nope, I was just saying, yeah,
Okay, yeah.
you
Yep.
wanted to be useful.
Yeah, exactly. All right. So I built that tool by building on the existing single cell sequencing alignment tool called Callisto. So Callisto was built in the Pachte lab, and it's still very widely used to analyze single cell RNA sequencing data. But basically, instead of the standard cell by gene matrix, I wanted to use Callisto to align to the PalmDB so that we would get a cell by virus taxonomy matrix, so that for every cell, I would know which RdRPs were present in that cell. And like I said, they have estimated taxonomies associated with them based on the clustering. And maybe this is something that is more familiar to you guys. Usually I need to spend more time with the biologists on this slide, and I feel like this is going to be the opposite. But just one little detail about the algorithm that is kind of cool. So Callisto is a nucleotide aligner. And if you want to use a nucleotide aligner to align nucleotides to amino acids, you can either reverse translate the amino acids into all possible nucleotide sequences, encoding them, but then you end up with a huge index because it becomes a combinatorial problem because our genetic code is redundant. So for every amino acid, you might have several triplets encoding that, and then you need to make all possible sequences based on those. So instead, we came out with our own non-redundant genetic code and then did the alignment in that nucleotide non-redundant genetic code space. And the code that we chose to do that is called a comma-free code. Basically, a comma-free code just says that you can only read it in the correct frame. So let's say we have our triplet code, so all words are 3 characters. We have 4 nucleotides, so 4 possible letters. So let's say the word ACC makes sense. It encodes an amino acid, and ACA also makes sense. Then CCA and CAC cannot make sense anymore. These are nonsense triplets. They cannot encode amino acids anymore. And that's a mathematical theorem. You can solve for how many possible words you can make in a comma-free code. And it turns out for triplet words with 4 possible letters, that's exactly 20. You can make exactly 20 possible words in comma-free code. And 20 happens to be exactly the number of amino acids. And so as a result, in 1957, when Francis Crick from Crick and Watson, the discoveries of DNA, Crick at the time knew that there were 4 nucleotides and 20 amino acids. And so he hypothesized that our genetic code was a non-redundant comma-free code because you happen to get this magic number. He was wrong at the time. They later got it right. But it happens to work really well for our algorithm here. So we made a comma-free code. That's what's one of the many things that are happening under the hood.
Yeah, no, let's talk about that at the end.
Okay.
We'll discuss it again.
Okay, great. And yeah, so now we can use Callisto. This is work together with Delaney Sullivan in the Pachte lab, who at the time was completely rewriting Callisto, so it's much faster now. So we work closely together to implement this. And you can actually use it for any amino acid reference. I'm using it here with the PalmDB to get my cell by virus matrix. And I also just wanted to emphasize again that we still also get the standard matrix of cell by host genes. That's just the standard workflow. And so we're adding this additional layer on top where for every cell, we know the host genes And now we have some indication of which viruses were there. All right, so we did some benchmarks and validations. I'll skip through this because I think I might run out of time. I'll also go kind of fast through this so we can get to the more interesting results. The main thing here I just want to talk about, so Hannes, you already mentioned the majority of the sequences in your sample, if it's a human sample, are going to be human RNA. And so you need to be very good at distinguishing viral versus human. And that can be a challenge, especially sometimes, reference genomes are contaminated. This is less of a challenge here since we're only looking for the RdRP. But I just wanted to briefly mention that in a paper, we describe and we implemented 8 different workflow of doing something called host masking, where you try to separate host and viral. And it turns out that's actually quite complicated, and you can get totally different biological results depending on how you make decisions under the hood. And I don't think we solved this problem. I think the main takeaway here is just be aware of what your algorithm is doing so that you understand how these decisions are made. So I'll skip through some of this. Basically, one idea that we try to introduce is that most other workflows, you just do a very conservative alignment to the host, and then all of those sequences get thrown away, and you end up with like few counts for your virus. However, we found that even confirmed viruses usually have some sequences that also have some overlap with the host. And so we were trying to introduce this concept of we'll keep track of all of the sequences. We'll mark the ones that also align to the host, but instead of filtering reads early on, you filter viruses later on, and you can try to make a more informed decision for sequences that are ambiguous, whether they are more likely to belong to the virus or more likely to belong to the host. I really think this is very early work. I don't think we've really flushed this out well, but this was kind of an idea we were trying to introduce in the paper that I hadn't really seen elsewhere. All right, so we applied this method to a data set of macaques that were infected with their Ebola virus. And so basically, these monkeys were infected with Ebola virus, and then their blood was sequenced over the time course of Ebola virus disease. And in addition to sequencing the macaque blood, they also did a spike in, so basically added a little bit of a cell line cells, so that in your sample you have the macaque cells and you also have the cells from the cell line. They're basically like a negative control, and I'll get back to that. That's actually really important. All right, so basically what you're seeing, if we analyze that data set with our data, is that first, let's look at Ebola virus. We already knew Ebola virus was there, so it's just a positive control for us. But what we see is even if we're looking just for the RdRP instead of the entire genome, we get overall low counts, but the relative abundances are still, like the relative trends are still captured. The relative trends are still correct. So at baseline, the monkeys don't have any Ebola virus. And then during incubation, so when they get infected with Ebola virus, you see this spike. And then it drops down again. And then over the course of the actual Ebola virus disease, the counts of detected Ebola virus RdRP increase again. And because we know the host gene profile of every cell, we can assign a cell type to that cell. And so what we see is that Ebola virus is mostly in macaque monocytes. And so that makes sense. So that's where Ebola virus should be. Okay, but we already knew that Ebola virus was in these cells. So what other viruses did we see in those cells? This plot, every dot here is a cell, and you can basically read this as the number of different viral species detected in every cell, or different RdRPs detected in every cell. So you see this slight increase towards the later stages, in Ebola virus disease, and that's mostly driven by an increase in neutrophils at these later stages. And that could be because neutrophils like to phagocytose the environment, and so they might be picking up the other viral RNA through doing that. So overall, we identified over 10,000 RdRP-like sequences. So what are all of these virus-like sequences? This is where the spike in comes in. Turns out most of the virus-like sequences that we saw are equally present in the cell line spike-in as they are in the macaque cell. So that probably indicates that this has nothing to do with the macaque or cell line. This is something that was introduced during the process of sequencing. And so it could be a contamination of laboratory reagents, could be some sort of environmental contamination, but it actually turns out that a lot of these sequences, we can find them if we just sequence raw laboratory reagents. So blank laboratory reagents contain a bunch of viral sequences. We are not the first to report this, but we can kind of see the same sequences just across totally unrelated experiments. This is just sequencing blank reagents. This is a new method where they introduced a spatial technology based on Illumina sequencing. And so mostly my takeaway here is that we do need to be very careful when it comes to interpreting the results that we're getting from this workflow, because not every sequence is actually going to be indicative of a viral infection. All right, so how can we try to narrow it down more towards which of these viral sequences or virus-like sequences do we actually care about? And so one of the things that we can do if we have this single cell resolution is we can look at which sequences are just kind of equally everywhere versus which sequences actually show some type of cell type specificity, like you would expect from a real virus. Real viruses are not usually just kind of omnipresent in all cells. And so, for example, I'm just showing two examples here. One is our Ebola virus, which was mostly in monocytes. And then the other one was this estimated alpha coronavirus species, which we saw mostly in neutrophils. I don't think this is necessarily indicative of this sequence infecting the neutrophils. Yeah, Hannes?
How do you find out during sequencing what cell your sequencing comes from? And do I now have to do sequencing for every cell type to then have this method working?
Yeah, no, great question. So this is done based on the host genes. So we basically have they're called cell atlases, where we have specific marker genes or gene profiles based on which we try to assign cell types. And depending on the sample and the organism and the cell types, this works better or less good. For blood, the cell types are pretty well characterized, so it's relatively easy to make cell type identifications. But this is it's a huge ongoing discussion still in single-cell RNA sequencing is cell type assignment, how to do it correctly. And yeah.
Okay.
Great question. Yeah. And then we have some sequences that are just kind of equally everywhere, just kind of floating around. And so one of the more interesting analysis I think that we were able to do is that for the sequences that had this higher cell type specificity, we were able to predict within the same cell type whether the virus was present or not based on the host gene expression. So, for example, we could take all of the neutrophils, and then just by looking at the host genes, we could correctly predict which of them would have a virus-like sequence mapping to this alpha coronavirus species or not. And then if you look at the highly weighted genes in those predictions, a lot of those are indicative of viral clearance. So that also makes sense. All of this you can only do if you have that single cell resolution though. All right. So overall, we now hopefully have a technology that I hope checks all of those boxes. We really tried to make the workflow as easy as possible. So this is one simple version of the workflow with one type of this host masking that I talked about. So including installation, it's just 3 commands. You install, you build a reference with host masking, and then you run your counting. So you analyze your sequencing data. You could do this on a laptop if you wanted to. So even if you do a single thread, you can analyze 1 million sequences in 1.5 minutes, and the memory requirements do not exceed those of any Mac. And like I said, the generated reference index is relatively small, so really very computationally efficient workflow. And then I also just wanted to point out gget, which is another software program I built that I'm using here to get our host genome, which is human in this case.
So what is this gget? Who developed it and what is it for? I've seen this a ton of times now appearing.
Oh, good, great. Okay. Yeah, let me show you more gget. Hold on. So, oops, wait, where did this go? All right, so you can find out more about gget by just going to gget.bio. And this is a gget website. I first developed gget. I'm still lead maintainer, lead developer, but it is an ever-growing project. So gget is a collection of these gget modules, and each of them has one very specific function that you often have to perform in genomics research. So this can be either querying of reference databases, like I'm showing here, for like Ensemble, UniProt, or NCBI. I had a gget AlphaFold module. I think now you should probably use ColabFold instead of this, but when it was really difficult to use AlphaFold early on, this was actually very widely used. There's a BLAST module that allows you to very quickly check what your sequence is most likely. Like you can basically check what does the sequence look like comparing to all known sequences that we have. Enrichment analyses, so what are my genes related to? A lot of different things. But yeah, basically you have all of these different modules. Everything is like very clearly documented, hopefully. And it's supposed to be really easy to use. So gget info, for example, is one of the most widely used modules, and it makes it super easy to get information about any genes across UniProt, NCBI, Ensemble, which otherwise you'd have to click through all of their different web interfaces. And it gives back everything in a human and machine readable format. And we had a huge spike of downloads for some reason. A lot of the AI scientists built on gget. So yeah, we have an ever-growing community on GitHub of users and contributors.
So it's not as like it's a collection of many tools, and the distribution of what it does is mostly like some sequence fetching, sequence alignment,
Yep.
like seed finding and genome annotation things.
Yep, exactly. Yeah. So I think the most important thing now so it has all of these functionalities that are very widely used. In addition to that, it gives a really great backbone to add new modules. So if there is a new database, for example, that is kind of difficult to get access to, I actually have some slides about one example. You can very easily plug it into this as well.
Yeah. Okay. I was not kidding. Like I now remember that you told me that you did some gget stuff at some point, but I think I just was at a broad or at broad presentations. It came up multiple times that someone was using GGET, and I was just wondering from
Oh,
that perspective.
great. Awesome. Yeah, good. Yeah, super happy. We now have some funding for maintenance as well. And up until now, I was just kind of doing it unpaid. But now hopefully I actually can pay myself and other people who are maintaining their own modules. All right. I'm super excited to hear all of your questions. So I'll stop here. I just wanted to thank my current lab, which is the Sabeti lab at the Broad. Most of the work I talked today about was done in the Pachte lab at Caltech. So these are all of the people that were involved in that. And then obviously my funders, especially Future House and the Eric and Wendy Schmidt Center at the Broad.
Cool, thank you. How, like, if we now want to use your this tool where you do your fetching of sequences for RNA viruses with this RNA polymerase, then would I now need to go ahead and take tissue out of my patient and then throw it into some microfluidics device and then do my sorting and then sequence and so on? Or how does it go?
So if you want to generate a new single cell RNA sequencing data set, then yes, that is the work that goes into that. I talked mostly about single cell RNA sequencing, but the method works just as well for bulk RNA sequencing. And so you can use it on any existing data set as well. So you could reanalyze a data set that is already deposited on the sequence read archive. We are analyzing it now for all of the sequencing data that we're generating in West Africa as part of the pandemic prevention work. Yeah, so I'm much more involved in the data analysis side than the actual sequence generation side. But the software itself, you can just pip install and then see.
Yeah.
It's easier if you have an existing sequencing data set and you just kind of want to see what RdRP-like sequences are in there.
Okay, but it's not easy to use this for like this sort of pipeline for diagnosing whether someone has a virus because that would be expensive to get the sequences.
Yes, so
Okay.
there's a lot of efforts to bring, we call it metagenomic sequencing into hospitals. So we are doing that in Africa, but like you said, it is still very expensive. It's becoming cheaper and cheaper every year, but it is still very expensive. And so actually a lot of the work that I'm doing now is building models for prediction based on clinical data to make more informed decisions around who should we sequence in the first place, because it is a very limited resource. And so there are more and more efforts toward that, but usually what is happening right now is we start sequencing when it's already bad, when it's already an outbreak, when we kind of just need to know, okay, this is clearly an outbreak. Is it Ebola or is it Marburg? And we're trying with Sentinel to get to a point where we sequence before that and we know beforehand. But yeah, we cannot sequence everyone. Luckily, we have some sequencing capacity, but we cannot sequence everyone. And so we're trying to make good informed decisions around who to sequence. And Pardis always says every negative test is a wasted test. Similarly, this is not just sequencing, right? You can apply the same to COVID tests. So we're trying to build models to do this hypothesis-driven testing where basically almost all tests should be positive and they're more confirmatory.
Okay. What else? Right. Can we go back to the slide where you do this comma code thing and
Oh,
then
yeah.
why like I don't understand this equation for how it gives us the why is it like that? Okay, no, first of all, like I understand that it makes if you say that A is your start, then the first three ones are sense, the second three ones are sense, and so on, and the rest is nonsense. What does this now have to do, or like, why do you call this a code without commas?
Oh, it's called a code without commas because you don't need commas to delineate the code. Because let's say Like
Okay.
with this code, like you don't need a comma here. Like you
Because
could
you know
add
that
a
it's always 3 long.
no, because I think it's not, I don't think it's super intuitive, but the idea is that, right, let's say you added a C here. Um, CAC, oh, actually CAC does make sense. Let's say you added a um a letter here, like a T here, for example.
Mhm.
Technically, it would be T, comma, ACC, comma, ACA, comma, to make sure you're still in the right frame. But because TAC is nonsense anyway, you don't need the comma. So it's a comma-free code because you can only read it in the correct frame. Doesn't matter if you start at the wrong point, you're gonna get nonsense unless you're in the correct frame.
I don't know. Like I thought that all our like we have. I thought that all 64 codons have some mapping to like either being a start codon or to being a certain amino acid.
Yeah.
So why do we call any 3-letter combination nonsense if all combinations have some meaning?
So in our real genetic code, that is the case. But because we have 64 combinations and like 20 amino acids, you end up with this redundancy. Um, and and if it were a comma-free code, you wouldn't be able to have this redundancy because you only have 20 nucleotides, 20 triplets. Um,
Yeah.
but this is not how our actual genetic code works.
Yeah, but it is redundant. So how could the guy, Francis Crick or whoever was it, make his inference that we have three nucle yeah, no, what's that we have three-letter
Triplet
code?
code.
Yeah.
Um, so he made that theory based on that you get this magic number 20. Um,
Yeah,
so
but I don't understand how we get the magic number 20 because it's
oh,
not the case that it is not redundant.
oh,
So
oh, wait, you get the magic number 20 from the comma-free code theorem. That has nothing to do with our genetic code. Like for
yeah.
any triplet code with any 4 letters, you can only make 20 words. And so there was this mathematician, Coulomb, at the time that just published on this theorem. And based on that, even though, I mean, technically Crick published a year before that, but based on his work, Crick just hypothesized, okay, I know I have 4 nucleotides and 20 amino acids. Uh, if it is a triplet code, like we get this magic number 20. So maybe, maybe it is a comma-free
Okay.
code with
So
triplets.
if Francis Crick applied wrong reasoning to get the correct results, because it is not correct that we have a one-to-one mapping between our codons and our amino acids.
Yeah. But so you're saying he was correct and that he thought it was triplets?
Yes, and his thinking that it's triplets is correct, but his reasoning was wrong. So he didn't.
Yeah,
Um,
his hypothesis was wrong. Yeah, his like he was proposing that maybe our genetic code was a
yeah.
non-redundant comma-free code, and then that was not correct.
Yeah.
Um, yeah.
Okay, I thought that this was actually like a correct argument for why we would have 20 amino acids. Sorry, for why it would be triplets, and like an inference that we can make for why it should be triplets. But yeah, I understood.
Sorry, I also just realized there's a lot of comments and
Yeah,
questions in
some
the chat.
questions you already addressed, like the graph stuff, and we're getting to it. But now the freshest one is, oh no, that's just a comment. Sydney Brenner discovered codons 3 nucleotides by applying frameshift mutations. Cool, cool. With respect to the cell type, how do you control for bacteriophages and if they are coming from a specific bacterial cell?
Yeah. Um, we don't. So I mean, we have estimated taxonomies that can tell us maybe this is more likely to be a bacteriophage. Uh, Based on the cell sizes, at least in single cell RNA sequencing, you are less likely to sequence a bacterial cell. But you can still have bacterial cells that are being co-trapped with the host cells or somehow infecting the host cells. And so we definitely do see bacteriophages in the sequencing, single cell sequencing data sets as well. Generally, I wouldn't expect to actually like have like sequenced a bacterial cell, just based on how the microfluidics works of single cell RNA sequencing. In bulk RNA sequencing, though, you would just have your bacterial RNA in there as well. Yeah, good
I
question.
guess the question from Timur we already answered, and if I understand correctly. And then why single cell sequencing with barcode resolution to a single cell is biologically interesting? I don't understand the question. If you do, then feel free to go ahead.
Yeah, maybe just basically what is a process where it is important that it's a single cell synthesis because you take a sample of a tissue and identify, whatever viruses, and where does the fact that it's a single cell sequencing, why is it helpful in this scenario, for example? Or maybe
Yeah.
there are different scenarios where it's useful.
Yeah. Yeah, good question. So, I mean, I think with regards to this technology in particular, one of the examples that I gave, right, all of the predictions based on host gene expression that requires single cell resolution, right? Like in our sample, let's see. Let me go through all of this. Right, like for example, this analysis or any sort of cell type specific analysis you can only do if you had single cell resolution, because otherwise all of these cell types would just be, and their RNA would just be jumbled together in a smoothie, and you wouldn't be able to separate them out. So I wouldn't be able to tell, you wouldn't see this difference. Everything would just kind of be everywhere. And then this analysis, we actually look even within the same cell type in individual cells, is there different gene expression if the virus is present? And so this type of analysis you couldn't do at all from bulk RNA sequencing, which is also done a lot. And so most of the diagnostic sequencing is bulk RNA sequencing, where they do exactly what you said, where they just take the tissue and they sequence everything. It's much cheaper to do. You get higher resolution data from that. So there's definitely pros and cons to both. But this is this like cell type separation and really looking at individual cells. It has a lot of biological benefits beyond what I'm doing here, but this is why people do single cell RNA sequencing.
So basically, you tag every piece of RNA differently depending on the cell, right?
Yep,
And
exactly.
then you need to classify cell types and maybe to the level of expression of individual cells.
Yeah,
Yeah.
so the cell type is classified based on the host genes. But yeah, you tag every single RNA molecule has a tag that tells you which cell it came from.
Basically on the expression profile. You
Yes.
classify. Okay.
Yes, and then the cell type is based on the host gene expression profile. Yeah.
Okay, thank you.
Yeah.
Yes, very interesting.
Great. But that's why I wanted to build on Callisto, because it has all the algorithm that you need to keep track of those barcodes.
Okay, let's have a last question from Furkan. Do you have insights with respect to codon aware versus amino acid based alignment in Callisto?
Could you tell me a bit more? Are you talking about?
Like, I think you had this, like we can either map to amino acid and then align the amino acids, or we can align codons, or I guess DNA sequence.
Yeah, you basically mentioned you could have all the combinations for a protein sequence in the nucleotide space and
Yeah.
then align
Yeah.
or
I see.
align on the protein space, basically. Yeah.
Yeah. So actually, it's such a huge combinatorial problem. Like it would be so computationally unfeasible We didn't even try that. Like even if you
Oh,
just
yeah.
have a few amino acid sequences, it's just not. It's not actually. It's not actually an alternative that you could do.
Does PalmDB not have DNA sequences as well?
They do, but you end up again with the trade-off where the amino acid sequences are nice because it's like a very clean data set where every sequence is like different enough that you can distinguish between viruses. You don't get too much interference. You know, if your reference sequences are too similar to each other, that also messes up the alignment because your read might map to like a lot of different references. And there's some ways to handle that, but overall, I found that it's better to use the amino acid space in this case. And even in the amino acid space, there's a lot of similarity between RdRPs. So that's why I'm operating in the amino acid space. But PalmDB also does have the nucleotide sequences. So one thing that we're exploring is, can we use those to build a GLM, to just train a model to know what nucleotide RdRP sequences look like, and then those can be applied directly to the nucleotide reads.
I see. Thanks.
Okie dokie, then let's call it a day here. And Laura, thanks for presenting everything. The recording is stopped here now, more or less. But the slides where you say confidential, I assume those are the ones we should remove from the talk.
That would be great. Yeah, if you could remove those from the public YouTube channel. But awesome. Thank you guys so much for all the questions. This was a blast. Thank you.
Yeah, thanks for coming here. And well, I don't know, what's your current plans, research, and so on? Are you now applying for faculty positions or stuff like that? And are you now focused on this Sentinel project 100%? So what's happening?
Yeah. So I'm research-wise right now, I'm pretty much focused on Sentinel. Good question about my career. I am pretty set on not being faculty, but I don't know what I want to do instead. And I'm having a lot of fun right now. I mean, the Sentinel project it's pretty huge. I'm going to Africa next month to start deploying some of the GPUs. The GPUs are being sponsored by NVIDIA, so I get to, talk to NVIDIA and Anthropic a lot. They're sponsoring a lot of my research right now. So all of that is really fun and cool. So I'm also not in a haste to go somewhere else. But if Bowles ever needs a computational biologist, let me know.
Okay. Not sure how far along that is, but at some point, I'm sure we're interested in some target discovery or things like that would care about. Okie dokie.
Congratulations, by the way, on the release. That's great.
Thanks. Then see you around and see everyone else around as well. Maybe next week, maybe some other time. Have fun, everyone.
Awesome. Thanks, Hannes. Bye.
Bye.